Yeah, so let's get started right away. I'm really happy that you invited me to give this talk. It's really an honor being a new member here. It's always really great to get to know many more people, and I of course want to get to know also more about what your research ideas are and what you're working on. I'm going to show you what I have been working on the last years, and also what I will be working on in the near future. And of course, given that I'm working in the Center for Biodiversity Monitoring, there will be some parts of the talk also about monitoring, especially from an agricultural perspective. So a brief outline of what I will show you today. I will show you a grassland trial that I was involved in at the University of Göttingen. Some of you may know this experiment, and we did work on insects in there. Then a crop rotation experiment together with members of FINROP, the Institute of Sugar Beet Research in Göttingen, an intercropping trial in Münster, and then finally some new research we are doing in Lower Saxony. And I hope that this will give you a good overview of how insects can be assessed, biodiversity can be assessed in farming systems overall, and how experiments can help to find out about drivers of biodiversity change. So how can we disentangle nature? Well, of course, like the left picture is an imaginary landscape with everything entangled. Let me use the pointer here. Everything a bit entangled here. And to disentangle that, as you may all be aware, a good way of disentangling that is to set up a replicated randomized experiment, as is often done in farming systems and agricultural research. And this is mainly what I will show you, but we'll come back to the landscape scale perspective at the end of the talk. I started off as a biologist, and I started off in grasslands. And one of these experiments I just briefly want to show you is this one here. It's already terminated, but it was running for several years in the Solling Mountains, so about in the middle of Germany, set up with the framework of a functional biodiversity cluster of excellence in Göttingen by Johannes Isselstein and colleagues. And you can see here that this is all a comparatively old grassland here, about 100-year-old grassland that was grazed and mown in the past. And what you can see is plots were established, and the different colors of these vegetation patches here comes from them being either fertilized or not, mown 3 times a year or only once per year. And also there was a biodiversity manipulation, being grass-specific herbicide was used, or herb-specific herbicide, to basically manipulate the functional composition of the sward. And looking at this in closer detail is here you see herbicide application against herbs leads to a grass-rich sward, whereas you have a herb-rich sward on the left-hand side here. This is not the same experiment, but it's close by, and you can see the difference in great detail here. And what we looked at in these grasslands, so we went to each of these plots for several years. We sampled insects with a wide range of different methods. And I'm only showing you one result here, which is this one here. We looked at these little leaf hoppers that are present in high numbers and high species numbers on these grassland patches. And this is the richness of the leaf hoppers, and this is the abundance. The kind of diagram you're looking at here is a structural equation model that you may all know, either you've seen it already or you know it from the intelligence quotient, for example. The intelligence quotient that is used in psychology is also basically a structural equation model. And what you can see here is relationships among different variables. So these two blue ones here are the ones that we measured. And these ones down here are the experimental factors. And traditionally, you ask the question whether, for example, fertilizer application would affect richness and abundance of leafhoppers, or cutting frequency, or graminoid enhancement, which is this kind of herbicide that reduces the dicots. And what you can see here is that there are only a few direct effects on the leafhoppers, but also lots of indirect effects that are mediated by either vegetation structure or plant diversity. So this shows that grassland management, these kind of three treatments here, of course have an effect on vegetation structure that also affects the richness of plants in the sward, and then you have these effects on the leafhoppers. So this shows that replicated experiments can be used to look at interactions between management, vegetation, and insects. And the main path here is, for example, that cutting frequency has kind of positive effect on leafhopper abundance. So if you keep the sward short, you have higher abundance, which may be due to the fact that they like to have a bit warmer microclimate, being ectotherms. And if you have more abundance, you also have higher richness. So these are the strongest arrows in here. And then there's one from fertilizer application having a positive effect on vegetation structure, meaning higher biomass and higher height here. And that one here, the higher the biomass, the higher the height, the lower the plant diversity, but the higher the plant diversity, the higher the richness. So you can see that this is all like disentangled by this kind of model here. And you can spend a long time trying to understand it. The interesting thing is that it gives you a network perspective, and you start to think differently about these systems. Okay, the next experiment I'm going to show you is on arable farming systems. So that's the one we did together with the Institute of Sugar Beet Research in Göttingen, where a PhD student of mine has been working for several years. And the idea here is to have elements of a crop rotation being laid out at the same year in space. So each element of a rotation, oilseed rape and winter wheat and maize and so on, are present in parallel in the same year. And you can then try to look at effects of the preceding crop, and also you can compare crop identity effects at the same site. And this is what it looks like here. And wherever you have these kind of white rods, you have insect sampling devices. For example, we set out pitfall traps and transparent pen traps. So not the kind of yellow pen traps you would usually use, but we used transparent ones that don't really attract insects, but they more or less passively capture insects that move through the vegetation. And I'm only showing you one result here, which is quite interesting. The idea is to check whether a diverse crop rotation has positive effects on biodiversity. And you can see, yes, the more different crops you grow over time, the higher your abundance. In this case, it's beetle abundance. But this is a comparatively weak effect. So you get from 110 to about 130 beetles here. The much stronger effect is the crop identity. So if you grow oilseed rape or winter canola here, you have much, much higher abundance than if you grow, for example, sugar beet or winter wheat. So the crop identity here is much more important than the crop diversity. Okay. Moving on to another experiment. which is an intercropping trial in the framework of an EU project that we just finished with several sites across Europe. And in there, we manipulated the mixture, so basically monocultures versus mixtures of legumes and cereals. And of course, we also monitored insects, not only using these cameras, but I show you this photograph because it might be really interesting for you, that we are at least trying to use also such devices to monitor insect activity. And what you can see is that the richness of arthropods differs between monocultures and mixtures. So if you have either a wheat or a bean mixture monoculture, you have a bit lower number of arthropod species. And if you go to the mixture, you increase the species richness. And the low input system that is not receiving herbicides and not fertilized has higher diversity. However, of course, the high input system also shows this positive effect of mixture, and there may be interesting trade-offs between productivity or yield and arthropod diversity. If you want to look at the details, just check out this paper here in Basic and Applied Ecology by Jana Brandmeier. It's open access, so you can all read it, and there are some interesting further information on that. Also in that system, we set out these kind of structural equation models, and this is ongoing work by another PhD student of mine, Silvia Papagallo, and we put the yield up here. So in this case, the model includes also yield and crop diversity being the main manipulated variable. And in between is basically biodiversity and components and also ecosystem processes. And what you can see is that biodiversity and ecosystem processes are more or less uncoupled from the yield. So there is no direct path, for example, from crop diversity to yield that is supported by the model here. Only those ones that have a color here are significant. This is the low input system, and this is the high input system. And what you can see in the low input system, weed biomass, because there is no herbicide being applied, weed biomass has a negative effect on yield, of course, as everybody knows. In the high input system, this arrow is not there anymore because there is basically no weed biomass anymore. And you get some other internal things happening here. So organisms interact with one another, but independently of yield. So this again shows that in this system, it may be possible to attain high yields, but still have biodiversity in the system and to do crop diversification. Okay, briefly, I would like to show you something about landscape scale experiments. This is biodiversity monitoring in real world landscapes. And we are currently doing that in Lower Saxony, and we did it also before in the framework of another study. One of these approaches that we used was to set out sampling grids of, in this case, yellow pan traps in 1 by 1 kilometer landscapes to predict heat maps of pollinator species richness here. So this could be really interesting because it can be combined with remote sensing approaches. And I think also from a phenorob perspective, this approach should be also very useful and interesting for you. We did that in replicated landscapes. So there were a total of 10 of these 1 by 1 kilometer landscapes. And we sampled everything, not only arable fields. We sampled cereals, grassland, oilseed rape, but also forest, root crops, and corn. So the major land use classes that were present in this area. And by doing that, you sample biodiversity in proportion to the true abundance of habitat types or of land use types in an area. And well, what you get here is you get, again, for each land use type, the number of species of pollinators plus the standard error that can be quite high in some cases. And similar to the crop rotation experiment, you find strong differences between these different land use types. And I think such results are highly needed in current debates on biodiversity on a landscape scale and the role of farming systems, because farming systems, depending on the crop identity, can have positive effects on biodiversity. And of course, we all know it for grassland, but it's also true for other land use types. The last thing I want to show you is a current study that we are conducting right now. Also in Lower Saxony, we are covering whole Lower Saxony, apart from the coastal areas here. And the idea here is, it's a new project called Finca, where we work with paired farms, organic and conventional farmers that have their farms in a vicinity of several kilometers. And the conventional farmers split a field here, and 1 hectare here of one of these fields is herbicide reduced. So basically there is no herbicide and no insecticide is being used on that field. And instead of using herbicides, organic farming practices are used on that field. Basically mechanical weed control is used. And I cannot show you results yet, but I can show you pictures from yesterday. So my postdoc, Svenja Bench, is currently in the field setting up traps on 90 fields across whole Lower Saxony. So it's really a huge effort. And we are also using automated monitoring devices here. This is an audio device that records bat and bird sounds. These ones are trap nests for wild bees. And these ones are so-called vane traps that lure pollinators into the trap here, and they are emptied after several weeks, and we then look at insect biodiversity, trying to find out if this kind of pesticide reduction has positive effects on biodiversity. Yeah, so with that, I'm already through, because the 30 minutes are also almost through. I showed you a brief overview of some of the things we did over the last years. Grassland management experiments showing complex effects of fertilizer, cutting frequency, and sward manipulation. Crop rotation experiments showing that crop identity is really an important component of agrobiodiversity. We had a brief look at intercropping trials, and I briefly discussed about potential trade-offs between yield and biodiversity. And then we looked at landscape scale, insect biodiversity, so what can be done to upscale all these kind of approaches to real-world farming systems. And with that, I'm done. Of course, as usual, I have to thank many, many people, especially my former Münster Animal Ecology group. Some of these people have come with me to Bonn and are now working at the museum. Of course, the BFN, the County of Lower Saxony, are to be thanked for the Finca project funding. The EFZ Göttingen, of course, is greatly thanked for allowing us access to the crop rotation trial, and the European Union for funding the intercropping experiments. And with that, I'm really looking forward to questions.